Our next intern is Katy Shelley from Jupiter High, and she has another interesting story because originally she applied for the mechanical engineering track because actually she can disassemble and reassemble engines. That I found it very interesting. But actually after talking to her and finding out that she has a great interest in physics, that like I never understood anything about it, we made a match with the imaging core, and actually she became so skillful in just 6 weeks on how to handle this super expensive microscope that actually she even helped other fellow interns and other scientists taking the images for their project. So congratulations.
Good morning. I'm Katherine Shirley, and I'm from Jupiter High. And as Ilaria said, I was in the light microscopy core. During my time there, I had the opportunity to get to use the microscopes with the goal of imaging nanostructures below the diffraction limit. Now, what is light microscopy or fluorescence microscopy? Essentially, what you're doing is you're coloring in specific structures within a cell or your sample using a fluorescent dye or a protein. Later on, we use light in order to turn it on or get it to fluoresce. From that point, we pick it out and we look at the smaller structures. For example, a hair cell out of a cochlea. Now, light microscopy is very simple to use, and it's used widely by laboratories and other scientists in order to get the images that they like. Now, compared to electron microscopy, where technical skills and background in electron microscopy is required before you can attain that data, the process in electron microscopy also takes some time. Here in light microscopy, we can get our images in minutes, if not seconds. Now, we use fixed cells, but we can also use live cells. For example, we can have a neuron under our microscope, we can treat it with a drug, and then over time, during our experiment, begin to look at the structural changes that occur. As I said before, we're able to visualize small structures, but there is a limit. This is our diffraction limit. It works with our magic number 200 nanometers. Now, what does this mean? So if you see over here, we have a low resolution image. If we have a structure that is less than 200 nanometers in size, or they're closer than 200 nanometers, low resolution microscopes will pick it up as one large 200 nanometer size spot of light. But over here, with our high resolution techniques, we're able to distinguish between those structures. We can see, no, it's actually 80 nanometers in size, or wait, this is actually two different structures that were just close together. Now, we have cutting edge microscopes here at MPFI, which I'll explain in this slide. We have a standard confocal, which is the fastest to image, the easiest to lose, but compared to the other two, it will give us the lowest resolution. We have the AirScan, which reduces photobleaching, which is essentially where you have a fluorophore and it's exposed to light for too long or for a long enough time that it just begins to not emit any more fluorescence, or you won't be able to detect it. If you can imagine, if you sent like your clothes, colored clothing through the wash several times, the color would begin to fade. We're using algorithms in order to compute out-of-focus light into in-focus regions. So it's a little bit more difficult to use, and there's a little bit more time that has to go into it in order to get the image that you'd like. However, we can get 1.4 to 1.7 times the resolution compared to the standard confocal. Lastly, we have our STED, or Stimulated Emission Depletion Microscope. We're using lasers to basically turn off surrounding fluorophores and spotlight a single detail. Now, because of that, we're able to get finer structures out and more information. But as I said before, we're using lasers. So the power going into that and going onto the sample will begin to bleach your sample a lot at a faster rate. It is the most difficult to use because you are using lasers. There's several different parameters and adjustments that go into, okay, well, like how bright do I want the sample? How much resolution do I need? and the pixel size. So there is a lot of time that goes into it, but in theory, it is diffraction unlimited, so you will be able to see well below 200 nanometers. Our goal, or my goal for this internship, was to be able to figure out which of these microscopes would help us image better, or would be the best to image samples with. I worked with a variety of different samples that had their own different dyes, and then I imaged them individually on each microscope. At the end, I took the images that I had taken, and I drew a comparison between them in order to determine, all right, which one will work the best. The first of our samples was cytoskeletal filaments. Here we're using a green dye called Alexafluor 488. We see here with the stead and the confocal that we do have a lot of detail. We can see the branches of the cytoskeleton begin to wrap around and give structure to the cell. However, we can see them with more detail here with the air scan. We have this zoomed in image that emphasizes that. Here we have this small portion taken out, and we can see a lot deeper into the cell and a lot deeper into the structure. We can begin to see how the cell builds itself. Our next sample is a Purkinje cell that we got from Christie Lab. This is using a fluorescence protein called TD tomato. This is a little bit more, it was sensitive when it came to the stead. Unfortunately, this protein was not compatible with the lasers that we were using, and immediately it was photo bleached. So there was no detail that we could get out of it. Our goal was to be able to get an image with enough resolution that we could count the dendritic spines. As you see here with the confocal and the air scan, we are able to get to that detail where we can see the individual spines, but with the air scan, it was able to enhance that detail. As we see here in this portion, we can zoom in and we can begin to differentiate between the spines, and we can begin our counting process. Next is our cytoskeletal lattice. Here we are using two different dyes. We're using Alexafluor 594 and a star red dye. These are your far red to red channels. We wanted to understand how this cytoskeleton was building itself, and we want to be able to detect the individual components. Here, as we see with this confocal and the air scan, they do blur out, but here with the stead, we can begin to see the individual markers begin to pop out. In this zoomed portion here, we can begin even further seeing that there's more so a pattern where they begin to alternate and build off of each other. Here's another. If we look back to our confocal and our air scan, we can begin to see that, well, it was blurred out. We couldn't see where their locations were exactly. From what we've gathered so far, I was able to determine that the confocal was good if high resolution wasn't required. So if we had structures that were greater than 200 nanometers in size, then this was perfect. It would work well with your sample being able to get you the detail that you want and the information that you needed. It also worked well with samples that might be more sensitive, and you can image it multiple times without having to worry about photo bleaching. Next, we have our air scan that worked best with darker samples. We saw that it was compared to the stead with our cytoskeletal filaments and that 488 dye, it was able to take the out-of-focus light that would be lost by the confocal and then put it to the regions that were in focus. That way we could see deeper. It also reduced the risk of photo bleaching because we were using multiple computer algorithms in order to get the out-of-focus light in there, instead of using a lot of laser powers as we did with the stead. The stead worked best with bright samples, as we saw with the cytoskeletal lattice. It also gave us a higher resolution because we're using strong beams. But we learned so far that we cannot determine which microscope is the best simply on like which one provides us with the best resolution. It depends on the characteristics of your sample. It depends on the dye that you're using, whether it's sensitive, and just the resolution that you may need and the size of your structure. I'd like to thank my mentor, Dr. Nikolai, for taking me through this process and being patient for me as I struggled to learn with the different microscopes and how to use it. I also like to thank Ilaria for helping me prepare for this presentation and guide me through the internship as a whole, and also to the Brain Trust donors who, with their generous donations, allowed for this opportunity that I could be a part of. I'd also like to thank my fellow interns for adding to this experience and helping me have fun.
Super good job, Katy, also on pushing through this AC that is trying to freeze us. So I'm sorry for the air, but you rocked it anyway. Questions for Katy? Yes.
I'm sorry?
It's just a big compliment, basically.
All right, thank you so much.
Yes, Paul.
The question was, based off of my experience, which microscope was, are you saying the best to use? Oh, user-friendly. I would definitely say the confocal. The confocal took very few parameters and it was very it was very easy to learn to use. But yes, definitely the confocal.
Yes. Based on this gentleman's comment, when was the first electron microscope online?
Yeah.
So the question is about history. Like, do you remember, like the stud microscope? Do you know when it was invented? It's kind of recent, right?
Well, it got the Nobel Prize in 2014, but it was being developed in the late 90s and early 2000s. But yeah,
For the stud.
Oh, for the stud?
Others?
Yes. But the stud has just recently come out for commercial. Yes, in electron microscopy, you're using fixed cells, so you can't image with live samples, but here, even with the stud, we're using lasers, we can still image with live cells.
Yes.
Came in originally with an interest in engineering, because it's pretty great. But what have you gleaned from this project that maybe has expanded your horizons?
So the question was, what have I taken from this experience and how has that might have sort of affected the career path, so to speak? I'd say that it's taught me a lot more about physics in general. It's taught me to be a lot more science-oriented, and it's also brought a lot of my attention into microscopy as a whole. It's been very interesting to be able to learn how to use these microscopes and even begin to understand some of the physics behind it. So I've become a lot more interested in physics.
Thank you, Katy. Awesome presentation.